I see, I see. Let's start. So, hello everyone. Today I'm gonna talk about Journal Club series, focusing on the single cell foundation models. I introduced 8 different papers, and I think this presentation will be the general overview of the single cell foundation models and the related research trends in the bioinformatics scene. Before I deeply jump in, I'd like to mention the term virtual cell. I think a lot of people have been very interested in the virtual cells lately. So in the past, many people started from the experiments, make the data, and then make the hypothesis and compare the data and the reality, and then make new hypotheses, new experiments, and so on. So this type of conventional approach was popular in the past. However, the problem of this type of research paradigm is the hypothesis generation is very manual and individual, so that the resultant model is not 100% perfect because this model is based on manual and individual hypothesis. However, in the drug development or biopharmaceutical companies, where the industry, that type of manual model is utilized to make a decision regarding patient selection, drug development, pipeline, and so on. However, many people started to focus on the virtual cells or virtual model, in which the model construction and model execution are repetitively iterated. And for example, there's a kind of virtual cell at this point, and then they can simulate to the real world, and then the validation of different two, the virtual cells and the real world are repetitively done, so that many people are trying to make the virtual cells very rigorously and thoroughly. And if anyhow the virtual cell can be close to the real world model, then maybe the decision, clinical decision or drug development decision or other decisions can be more accurate. So there is a kind of paradigm shift in the bioinformatics scene. Even if so, there's no clear definition of virtual cell. However, in the perspective in the Cell paper, there is a kind of definition of virtual cell saying virtual cell is learned simulator of cells and cellular systems under varying conditions and changing context, such as differentiation stage, perturbations, disease stage, stochastic fluctuations, and environmental conditions. And there is a growing need for virtual cells. FDA this year announced that they will discourage the use of the animal experiments while encourage the use of organoid computational models and advanced in vitro assays. And I think the computational model exactly corresponds to the recent emergence of virtual cells. And there are two ways of reconstructing the virtual cells. The first type is the knowledge-based graphs. We can think of gene networks or pathways or signaling. The type of graphs can be utilized to simulate the world, meaning virtual cells. So I think the physics-informed neural network can also be an example of this type of knowledge-based graphs. And that is popularly done in different research teams, including Jasmine Fisher Lab, the university, in the England UCL lab. And another paradigm to realize a virtual cell is the single cell foundation models, which is test agnostic, so that it can be applicable to various type of tasks. So we can think of as gene corpus data, then we can also think of another architecture of large language models, including BERT, GPT, and so on. And that type of general language models can be pre-trained to fit into the bioinformatics test. So that is called pre-training in this type of single cell foundation context. And that pre-trained models are now fine-tuned for its downstream tasks. So basically in the single cell foundation model context, there are two steps, pre-training and then fine-tuning if needed. And that downstream tasks can contain clustering, batch correction, cell type annotation, gene network inference, multi-omics integration, perturbation prediction, and so on. And please note that the Geneformer and the scGPT are two preceding single cell foundation models. And very interestingly, Geneformer is published in Nature, while scGPT is published in Nature Methods. I want to tell you a very funny story. So scGPT was published in bioRxiv before the publication of the Geneformer. However, bioRxiv is preprint. And And actually, the official publication is, as you know, the kind of peer-reviewed journal paper, right? So I think the strategy is somewhat not good for the scGPT. If scGPT is trying to publish their own papers in the Nature or Science, then that would be successful. However, Geneformer does that so that they will be published in Nature. However, scGPT became the second single-cell foundation models. So that's the reason why one is in Nature and the other one is Nature sister journal. Bioarchive, I think you can easily find that. I think that is a kind of March, and I think the official publication of Geneformer is May or something, and then the official publication of scGPT is somewhat October, August something. So I think there's kind of I think in the between. That's my impression.
The old Geneformer was May 2020. Final. I think that was about the same time
Same time.
as the first.
I see. So
Maybe that's
I see. And I want to mention models in the later slides in the detail. And please note that most of the models, 6 out of 8, are based on transformer encoder structure, while scGPT and cell-to-sentence are based on the transformer decoder structure. And please note that when we use the transformer encoder structure, the pre-training may be masked language modeling or MLM approach, and also fine-tuning can be something like that. And also, when you are using the transformer decoder structure, you may use NWP, next word prediction, or next sentence prediction, something like that. I will mention that in the later slides. Please note that the fine-tuning strategy can be somewhat different from the pre-training strategy according to the downstream task types. And also, popular transformer encoder is BERT. So, for example, Geneformer and AIDO are utilizing BERT architecture. So, and also GPTs is also another popular type in the transformer. So that is also utilized in the cell-to-sentence. And I'd like to mention why the last one is not requiring the pre-training in the later slides. Yeah. So your question is based on the selection between MLM and NSP or transformer encoder versus transformer decoder. I think MLM is quite intuitive in the application of bioinformatics. So I want to mention this one in the later slide, but we can think of the tabular data. We are dealing with tabular data in the bioinformatics, and we can make some masks randomly, and then maybe we can train the transformers to predict the masked ones. So I think MLM is very well applicable to the tabular data, while NSP is not that well fitted into the tabular data format. Next word prediction. So I want to mention this as well. So the trend. So encoder architecture takes the inputs of raw data, gene expression at this scene, and make the embedding space. And the embedding space is the direct application of the downstream tasks. So, for example, we can obtain the embedding dimensional vectors, and maybe you can cluster, we can dimensionality deduction or cell type prediction, and so on. That is the principle of the transformer encoder in the bioinformatics. However, in the transformer decoder, actually the gene expression where the raw data should be somewhat converted into tokens. So that is a type of embedding components. And then that embedding components are now utilized to to reconstruct the original data. So that is the decoder design. And there are two sub applications of the transformer decoder. Maybe we can utilize the final output. That is a cell-to-sentence. And also we can also utilize the penultimate layer of the decoder. That is what scGPT is trying to do. So there's a tokenization of the given data. Yeah, so I think that can perform as a kind of embedding stuff.
We don't need to like really.
Yeah.
figure out stuff, but it's just like exactly. Sounds like they both have the potential block.
Yeah, that's right.
I think it's really completion task that they could also have the process attention on the size of attention. And, yeah, it's like I'm not 100% like clear what's the difference between equal, but yeah, but it sounds kind of interesting to see this like partition.
Yeah.
Yeah.
I will illustrate on this one in the later two slides. So maybe comparison between transformer encoder and decoder. And transformer encoder predicts a mask gene expression, as I mentioned. So this is an example of the persist paper. And here there is a given gene expression. And then there's a kind of mask vectors to that so that we can obtain the masked input. And the transformer encoder is now trying to predict the masked components. So so that we can, the weighting of the transformer encoder is adjusted to expect the masked components of the input so that we can obtain the somewhat biologically meaningful. So this is another example, but for example, we can think of in a natural language natural language processing context, its keyword can be now translated into embedding space with high dimensionality. So that is what transformer encoder is trying to do. And these embedding vectors are utilized for downstream analysis. And this is what the principle of masked language modeling, or MLM. And actually this this is for the gene panel selection algorithm, but the binarization is not necessary for the general context of the single cell foundation models. And also this transformer decoder generates a next generation expression. So this is what the next word prediction, where NWP refers to. And given a sequence of words, the model is trying to predict what the next word is. And here the autoregressive approaches are utilized. And I think in the application of the single cell foundation model utilizing the transformer decoder, there are two sub applications. The first one is the generated ones for the downstream analysis, which can be seen in the cell-to-sentence. For example, if the model input is T-cell in multiple sclerosis, then maybe the model outputs a type of genes, and that genes are really the outputs that the cell-to-sentence is trying to utilize for downstream analysis. And also scGPT is utilizing penultimate layer of the transformer decoder. I think this is not related to transformer. This is general diagram of the autoencoder. And I think the scGPT is trying to use the middle layer between embedding space and the outputs. And I think penultimate layer will be utilized for downstream analysis. And this is a comparison table between BERT, which is the representative example of the transformer encoder, and the GPT-4, which is another representative example of transformer decoder. BERT is developed by the Google AI, while the GPT-4 is developed by OpenAI. And BERT considers both left and right context of the input sequence, while the GPT-4 considers input sequence sequentially from left to right. BERT was developed with 1.5 billion parameters, 3 terabytes of training data, while GPT-4 was developed with 340 million parameters and 35 terabytes of training data. As I mentioned, BERT is based on the transformer encoder by using MLM, masked language modeling, or NSP, next sentence prediction. And GPT-4 was developed by the transformer decoder utilizing the NWP, next word prediction. Here, NSP can be exemplified as follows. For example, when the given first sentence and the second sentence, the transformer encoder is trying to answer whether the second sentence is the right next sentence to the first sentence. So here, the answer is no, because the second sentence is irrelevant to the first sentence, meaning the second sentence is not next to the first sentence. And the BERT architecture is utilized for text understanding, while the GPT-4 is used for text generation. And this is the comparison tables between AIDO diverse transformer applications in the bioinformatics. So there are 3 types: encoder design, decoder design, scGPT style, and the decoder design cell-to-sentence style. So the first type takes the input of gene expression profile with some genes masked, and it is utilizing the masked language modeling to predict the masked gene expression values as a pre-training objective. And it utilizes the hidden states to embed and represent the cells. And the output is not directly utilized. The embeddings are the main utility for the downstream tasks. And the decoder scGPT style takes the gene expression tokens, which is referring to the sequence of gene IDs and values, and then embedded that into the decoder input. And it utilizes the next token prediction, which is very similar to next word prediction, to predict the next gene expression token as the pre-training objective. And it utilizes the finer decoder hidden states for the representation of the cell. And the outputs from the transformer decoder is used for training, and the hidden states are used about for downstream tasks. And the last one takes the input of gene expression profile as the input for the pre-training and embedded them into the decoder input. And it uses the next sequence generation as a pre-training objective to generate the natural language sentence describing the cell. And it's not utilizing the representation or embedding of the cells. It just utilizes the direct outputs of the decoder design. Yeah. And these are some comparisons of each 8 model. And I'd like to mention that cell-to-sentence and CELLama are utilizing the gene list so that they are not susceptible to the batch effect. There are 2 variations in the bioinformatics: biological variations and technical variations. Biological variations are what we are interested in, while technical variations are often referred to as batch effect. Just because these first 6 models are using the quantitative expression, gene expression data, they can be susceptible to the batch effect. And please note that here Geneformer is not that significantly susceptible to the batch effect because it has its own functionality to be scalable. So there are 2 functionality in the Geneformer. The first one is the median normalization, and the other one is gene length-based tokenization, so that they will not be that susceptible to the quantitative gene expression data. And the yeah. So O means no batch effect, yes. X means no. Triangle means in the middle. Power, actually, this is the first script I want to mention. Yeah. And the availability of the metadata is also another important component when it comes to the pros and cons of each model. So the first 4 models are not using metadata, but TranscriptFormer, scMulan, cell-to-sentence, and CELLama can take the inputs of metadata for their applications. I think that is very good. That is very good characteristic because, as you know, some data have their own metadata. For example, biological status, gender, birth, perturbation, and so on. And that type of metadata are applicable in the various unstructured data sets. I think if they are available anyhow, then maybe the downstream tasks based on these models can be more accurate according to because this is what the biological prior is meaning for, right? I actually benchmarked that. Yeah, actually, the reason why I mentioned this type of model is I have experiences to utilize them. So I use the tabular sapiens, and I use them for cell type prediction task. I think cell type prediction can be done without foundation model, something like SCVI can also do that. But I think it's very good benchmark because there are a lot of data sets and very popular benchmark testing. And here, I think I found scGPT, Geneformer, UCE, and CELLama have decent performances, while TranscriptFormer and two-sentence has moderate performance, and AIDO was worst. And please note that scMulan was not applicable in my execution environment. I think it's hard to. Yeah, I think so. I mean, the installation, I think, yeah, there's some issues in installation in the usage. And the time complexity also another important factor to benchmark its model. And AIDO was worse. And you see TranscriptFormer has middle time complexity. However, even if this is decent in the, for example, 500 seconds or 1000 seconds, it's not applicable to a lot huge data sets. But in the future, maybe we are dealing with a lot huge data sets. So in such a case, the scalability is very important feature. And just because they are not that perfect in the time complexity, I think they can have disadvantages in the sense. However, the scGPT, Geneformer, and CELLama has very good time complexity. Very good speed, computational speed. And please note that the reason why scGPT and Geneformer are so fast compared to other models, maybe due to the CUDA requirement. So the other models are not using GPU explicitly. However, scGPT and Geneformer requires to do that. So I think the CUDA requirement somewhat important in the time complexity. And also, the reason why TranscriptFormer can utilize the metadata is the tokenization is based on the context-based, which means contextualized gene embedding. So even if UC and TranscriptFormer are very similar to each other, I will mention that in the later slide. TranscriptFormer is somewhat advantageous regarding the metadata availability by utilizing the CGE tokenization. Yeah, so in this benchmarking, I use just pre-training data. And please note that I think TranscriptFormer or one other model are problematic to do fine-tuning. There is a problem. For example, please note that TranscriptFormer is research prototype level code. So we should have manual wrapper functions. And there are some other problems in the installation. I will mention that in the later slides. I will not remember that every detail, but I mentioned that. So there's an installation issue, fallback issue, I think flash fallback issue. So the fine tuning was not available for some models. Yeah, that's right. And these are the dependency tables of each model. And please note that TranscriptFormer is based on 3.11 Python version. Please note that the latest Python version is 3.13.3. So as you know, 3.11 is close to 3.13. So many functions are not incorporated into the TranscriptFormer because, as you know, what we are using is somewhat based on lower version of Python. I think there is a kind of problematic point I found. And as I mentioned, the CUDA availability is only explicitly required from the first three models. And as I mentioned, the flash fallback issue was there in the AIDO. Just because of that, I think the GPU availability is not perfect. And also the pre-training or fine-tuning of that model was not also applicable. So when it comes to the benchmarking, I use just inference from the given pre-trained model. It is possible. However, for example, fine-tuning or GPU applicability, that I think those are related to the flash fallback issue. I found that issues a lot of times when I use AIDO. So I think, I mean, as far as I know, this is related to the GPU, GPU or training issue in training a model that is required. I think these kind of functions were packages. So for example, when I execute the AIDO, I execute from the installation of some packages related to the flash fallback. So I think I use another packages to utilize the inference of the pre-trained model instead of the direct application using flash fallback. That's what I'm understanding. There's a lot of any troubleshootings. Yeah, so I mean, please note that even if the first three models are used, but the speed, type, time, complexity of the AIDO is significant. I think that is partially because of the issue of flash fallback. Also, as I mentioned, the TranscriptFormer requires some manual wrapper functions, including 4 different modifications I will introduce later. And one of them is also related to the flash fallback issue. And scMulan was not applicable in my environment and in my experiences. And please note that in the academic scene, the license is not that important, but in the commercial scene or industry scene, the license is somewhat important. And actually, out of the 8 different models, AIDO says they have their own copyrights, so that you are not using that. You cannot use that for your commercial use and so on. I think for bioinformatician, we should always be aware that our models should also declare what type of license we are standing for in the GitHub or other archiving sites. And from now on, I'd like to mention pros and cons of each other model. scGPT is fast and it has a good performance. However, it is susceptible to batch effect and the metadata is not available in the scGPT. And as I mentioned, the CUDA requirement can be regarded as a double-edged sword. It can support very fast computing time. However, the requirement is somewhat problematic in terms of installation or execution. I think even if I classified it as a cons, I think it can be regarded as a double-edged sword. And please note that I think the scGPT is utilizing the penultimate layer for embedding of its cell. And actually, scGPT is based on the single cell applications. However, very recently, I think sometime this year, scGPT Spatial is announced, and where the spatially aware fine-tuning strategy and the spatial transcriptomics applications were introduced. And there are multiple models in the scGPT, including whole human, whole brain, whole blood, and so on. And Geneformer, I think the good advantage of the Geneformer compared to the scGPT is the weak batch effect. I mean, as I mentioned, it utilizes the median normalization and gene ranking-based tokenization so that it does not explicitly utilize the quantitative gene expression. so that it can somewhat alleviate the issues of the batch effect. And also it is fast. It has a good performance. However, the metadata is not available. And I think many people have said in the X or other social network that Geneformer has a limited downstream performance. So which means zero-shot setting is not effective. So fine-tuning is required for every downstream task. This is a benchmarking between diverse models. I just showed those 3 of them in the bioarchive. And here, as you can see, scGPT can be applicable to many other downstream tasks, while Geneformer is only applicable to the time annotation and gene function prediction. Just because the Geneformer requires the fine-tuning from the pre-trained models. I think this is somewhat laborious. And so no, this is the only part, but
what they did. I think that's kind of like one biggest gap when like thinking about applying transformers to doing expression data, because like I mean there's some gaps. Expression is kind of a matrix, and longer there were sequences. Like this, like how they tokenize the embedded one of the like immediate thing that I'm curious about. And, yeah, it's like after
yeah, that's right. Yeah.
So which device you use? Although it's your laptop or the online service?
Oh yeah, I'm using different environments. For example, there is a US university. I mean, there are a lot of it's kind of cloud platform. I mean, I will mention that in the Slack channel if you want. I mean, just beyond the
That's doing the best of the ones you tried.
yeah, on this specific data without fine tuning. So yeah.
Well, sorry to interrupt. I just
Okay.
interesting slide.
Yeah. So I mean, I'm using the Great Lake. I think it's good, but also beyond that, I'm also using other platforms. So there's kind of, for example, University of Texas or California, University of California. I think we can use that freely available, and also yeah, I think it's part. Yeah, I will mention that after this slide. Yeah, there are a lot of, resources I'm utilizing. It depends. For example, I'm using local environment if R package is there, but there's no R package at this moment. But
I'm also curious that this are all language based on language.
language models.
So like this foundation model are language.
Yeah. So basically, as I mentioned,
Yeah.
Geneformer, AIDO, they are based on BERT. BERT is very popular language models. GPT-2 is the lower version of GPT-4 or 5. Mini-LM is a sentence embedder. This is also a language model. Even if the other 4 are not mentioning what type of model they are using, I think they can also be very similar to other famous models for sure.
How is that?
As I mentioned, transformer encoder is very easy to understand. And if we input a certain sentence or word, we can obtain vector. So for example, there's a cat, there's a sentence, there's a cat. If we input that in the, for example, sentence embedder mini LM, then we can obtain 384 dimensional vectors from the sentence. So that is what transformer encoder is doing. And I think transformer encoder is very intuitive in terms of foundation model approaches or downstream tasks. And actually, the last one is basically using that type of principles. directly because it does not require pre-training. These are 7 models are requiring the pre-training step because the general language models are not task specific. They are general, right? So these 7 models are somewhat modified to fit into the bioinformatics scene. However, the CELLama, this is using the just general sentence embedder, does not require pre-training, but even applicable to the bioinformatics context. So I think I want to mention that in detail in the later slides. So Geneformer, yes, Geneformer is based on BERT architecture. And I read the GitHub, this is a kind of off topic, but last year, journal club class, I read the Geneformer in detail, very detailed, including GitHub repo and the revision documents, as well as the paper or methods. And I think Geneformer is written in a very clear code. So very clear. So there is no flow of that, just based on my perspective. And it has multiple models, including 16 million parameters, 104 million parameters, and 10 million parameters, and so on. And AIDO, I don't believe, just for me, I couldn't find any advantage from this model, but maybe there is some. But it has disadvantages, including the susceptibility to the batch effect, unavailability of the metadata, low accuracy, and very, very, very slow speed, and the CUDA requirement, GPU memory error in some cases, and flash fallback issues, and license issues, and so on. And please note that this tool was developed by the GeneBio AI and hosted by Chan Zuckerberg Initiative. And the AIDO.Cell is an example application of AIDO.model generator. And this is based on the BERT architecture and has 4 different models, including 3 million parameters, 10 million, 100 million, and 650 million, and so on. And UCE, Universal Cell Embedding, has a good performance, and it incorporates diverse information, including chromosome information, RNA expression, ESM2 protein embedding. However, it is disadvantageous in that it is susceptible to batch effect. The metadata is not available, and it is limited to the protein coding genes. However, I do believe, for example, long non-coding genes can also be applicable to embedding of cells, but the UC is only utilizing the protein coding genes. I think this is another one type of limitation of the model.
When they train, essentially have like just 10 million cells by ourselves, and then train, basically use this architecture, and like we got tons of GPU hours to train by ourselves. Do you think it's like essentially generalizable to like other genes, or is it like a training issue, or is it more of a
Yeah, I think, as you mentioned, it is generally applicable to other models for sure, or other genes for sure. However, just because it is based on the protein embedding model, which relies on the real protein data, I think maybe they don't want to use non-protein coding gene. So but I think it's applicable. Out of distribution generalizability, I think it's possible.
or I think
Yeah.
it's like 70%. So
Yeah, it's still good. And TranscriptFormer is also very similar to UCE, Universal Cell Embedding, but the availability of the metadata is a very different point of the TranscriptFormer and the UCE. And also it supports the multiomics, including RNA expression, ESM2 protein embedding. However, it doesn't incorporate the chromosome position. And however, it is disadvantageous in that it is susceptible to batch effect. It is slow. It requires higher Python version so that it may lead to package version conflict. And manual wrapper functions are needed to use the TranscriptFormer because it is at the level of the research prototype. So for example, automated ensemble mapping, safe merging of config.json with fallback issues, and automatic supplementation of missing some columns, automated handling of duplicate genes and NA values, and a built-in end-to-end classifier are all required when I use that model. And also, just because it is based on the ESM to protein embedding model for cell tokenization and gene tokenization, it is also limited to the protein coding gene. And it is trained using 112 million cells, which corresponds to 1.53 billion years. And it has 3 different models, TF metazoa, incorporating all 12 species. I think the very characteristic of TranscriptFormer is it can cover many species. I think the general applicability to other species is also one clear paradigm of the single cell foundation models I could find. And also TF exemplar is applicable to human and 4 different model organisms, and TF sapiens is only applicable to human. And even if UCE and TranscriptFormer are very similar to each other, they have kind of shared features and distinct features. For example, they are common in that non-zero counts are used as genic, and also protein coding genes are only utilized for ESM embeddings. And please note that it is further incorporated into Saturn algorithm, which is cross-species single-cell integration method. And also, we can think of multiple proteins from a single gene, which is called isoforms. And that type of multiple proteins are also considered by using ESM2 embeddings and average it to represent a single gene. However, UCE is distinct in that the metadata is not included just because UCE intends to make universal cell embedding. So for example, as you know, metadata is very case-specific, researcher-specific, and also there are a lot of chaos in the metadata, unstructured data, and so on. So if the metadata is incorporated into the single-cell sequencing data, the embedding will not be that generally applicable. So the UCE better focus on the general embedding just by using tabular data only. And also UCE utilizes the chromosome information. However, in the TranscriptFormer, utilizes the contextualized gene embedding to incorporate the metadata. However, it is problematic in that it is at the level of the prototype level code necessitating manual wrapper functions. And I want to mention scMulan. scMulan takes the input of gene, actually all of the genes expressing more than 0. And then their corresponding values and some metadata and some task type. And as I mentioned, scMulan is a type of transformer encoder. So by taking that information, we can obtain such a embedding vector that can be applicable to downstream tasks. I personally believe that type of massive inputs are not good. For example, I mean, that's my personal perspective. When I use Llama 4, when I input, for example, 100 genes or 200 genes, and it said, oh, there are a lot of list of genes, I could not process that repetitively. So I think language models are not good at processing such a huge amount of gene lists, but scMulan encourages to do that. I was suspicious about the scheme. However, it insists that it has a good performance in the integration. So the metadata is available. I think this is a very good point. However, it is susceptible to batch effect and it is unable to run just in my environment. And for example, the input type is as follows, or the non-zero genes are used as input. That is a characteristic of the scMulan. And cell-to-sentence uses the inputs of a number of gene lists only, so that it is not that susceptible to the batch effect. And then there can be some metadata, including cell type information or tissue information, disease information, or tissue plus disease, and so on. And that type of tokens are provided to the cell-to-sentence so that the finer trained models can perform produce gene list from the cell types and disease, and also predict the cell types from the gene list, and also complete a similar gene list from the given a few cell a few given list of the genes.
This one, like you can directly do this task with local, but essentially try to train about the meaning. Give a few examples.
Example of this one?
Yeah, I mean, it's like it doesn't generate this am I correctly, and like in the input and the output, it's kind of like it's they're all attached.
Yeah, that's right. So cell-to-sentence directly utilizes the outputs of the decoder, transformer decoder. So for example, there can be some hallucination. For example, even if we designate some prediction portion, the outputs are not related to this type of format. That is a kind of problem. And also, I think just because of the reason, cell-to-sentence was pre-trained, GPT-4 or other general language models cannot do that properly because of the hallucinations or something. So there are a number of pre-training steps to fit into that type of data format we are deciding for. Yeah, I mean, the authors say so. Yeah.
This capability, like multi-omic integration, like perturbation prediction, a lot of things like that. It seems like the self-sentence is kind of potentially limited and with like downstream kind of big.
That's right. That's right. Yeah, I know her.
Saying that large language models which I definitely agree. I mean, I mean, I would say it depends. The perturbation, I think it is. Yeah, this one very few. I think that a lot of models, they don't perform like linear or additive model. I think
Yeah, I think I agree that. So as I mentioned, there are two different subtypes of the transformer decoder applications, and I think scGPT is more strategic compared to a cell-to-sentence because it uses the embedding information, so that is very flexible in terms of the downstream tasks. And cell-to-sentence has no batch effect because the only gene list is utilized and the metadata is available. However, it has a low accuracy in my benchmarking, and it has lots of gene expression information because it utilizes gene lists only, not quantitative gene expression values. However, the author mentioned that the results show that 0.815 variability is still preserved from the gene list. That when they are using regression models to reconstruct the gene expression from the gene list, they can verify 81.5% of the variability is still preserved. And there are three models, 160 million, one at 410 million, and 1 billion parameters. And it is based on the GPT-2 architecture. And the author utilized 100 highest expressing genes. And various applications are introduced in the latest version of the cell-to-sentence C2S scale, including the spatial transcriptomics application. And also, and the C2S scale, it introduced a fine-tuning strategy using reinforcement learning. And I'd like to mention the CELLama. So it uses the highly expressing genes, so similar to the cell-to-sentence. And please note that if we are using highly variable genes or differentially expressed genes, then we may need to perform the comparison of a cell to other cells, and that may hinder the scalability of the application. However, if you are using highly expressing genes, then maybe the calculation is much more simple. So just because of the utilization of the highly expressing gene can also promote the scalability of the algorithm, and the input of the sentence is something like that. The top genes are several genes of the cell, and also the context can be added to the sentences. And then, as I mentioned, mini-LM-based sentence transformer is there so that these type of prompt is input, and then we can obtain the embedding vectors. More specifically, mini-LM can support 384 dimensional vectors from the given sentence. Even if it doesn't require pre-training of the language models to fit into the bioinformatic context, I think this is applicable to, for example, data integration, cell type prediction, and so on. For example, here, just because metadata is given, we can find the clustering patterns according to organ and tissues. And I'd like to mention that there's a kind of mention that within its single cluster, there is a biological variation of cell types. So even if the metadata is given, the cell type variability is also preserved within its single cluster. And it has advantages in that it has no batch effect. It can avail metadata, and it has a high accuracy and fast, and the pre-training is needed. Many researchers mistakenly assume that general language models would treat gene names as unfamiliar words and thus fail to capture biological patterns. But I feel like general language models can also understand gene lists if they are given 10 or 20 items. And it has a disadvantage in that there is a loss of information because it uses the highly expressing gene lists, not quantitative gene expression values. Please note that this is based on the mini-LM, the sentence embedder, and also the fine-tuning strategy is introduced by using cosine similarity of the embedding vectors. For example, if cell I and cell J are within the same cell type, then the resultant cell embedding should be similar to each other. And also, if they are different cell types where different metadata are given, then maybe the embedding should be, the cosine similarity of the embedding should be lower or even zero within the different contexts. So the sentence transformer can be somewhat fine-tuned to make that embedding to satisfy that conditions. And I'd like to mention that Jeffrey Hinton, the Nobel Prize laureate in physics in 2024, once mentioned in his Nobel Prize banquet that this new form of AI excels in modeling human intuition better than human reasoning. So I do believe the general language models or transformer architectures are very good at intuition. So as I mentioned earlier, for example, input of thousands of genes are not good at, not encouraged to when it comes to the use of the general language models. And also even the general language models can also have an intuition to utilize the bioinformatics context. For example, the left part is my intuition. When CD19 is given in the first 5 differentially expressed genes or first 10 highly expressing genes, and I could think of it as a kind of typical B cell. And also S1, 0, 0, 8, 8, and 9 heterodimer are given in the bulk RNA sequencing data or single cell RNA sequencing data, I can think of, oh yeah, there is a neutral field activity. And also CON1A1, COL1A2, COL3A1 are given. I think, oh, they are activated fibroblasts. And also FLH1 and KLK3 are given. We can think of, oh yeah, there's a prostate cancer epicellular cells are there. So there's another example of the evidence that the general language model has such an intuition is the GPT cell type, which is also published in Nature Methods. And here, just because we are using top around 10 gene lists, GPT-4 already can infer the cell types from that. So even, the other seven single cell foundation models assume the general language models are not good at understanding the bioinformatics context. I think there are several evidence that the general language models can also understand the bioinformatics and also have an intuition to perform the downstream tasks as well. So that is the reason why the CELLama doesn't require the pre-training. And these are for take-home messages. Single-cell foundation models are preferred to be transformer encoders. GPU is not explicitly required for many foundation models, leading to higher user friendliness, but slower inference time. And each model has its own pros and cons and characteristics regarding the metadata inclusion, fine-tuning strategy, and practical applications. And also, general language model can also be employed for single-cell foundation models without pre-training. And I want to mention two perspectives, and this is the last slide I mentioned. TranscriptFormer initiative, but that many other researchers are now focusing on the foundation models to provide the flexibility and also scalability. And I think the very motivation of the TranscriptFormer initiative is to make the virtual cells to simulate the real cells or organs or patients so that they can facilitate the clinical improvement or drug development and so on, or pharmaceutical industry. And also, Demis Spitz, the CEO of the Google DeepMind, also mentioned we want to build a virtual cell. I think this is the ongoing paradigm and trend. Actually, I want you to be keenly follow the type of trend. Thank you.